Now we'd like to introduce one of the most important figures in pancreatic cancer research. Dr. Daniel von Hoff of TGen is one of the world's foremost experts in the field and has developed a number of clinical trials that are moving the needle in the fight against this disease. Dr. von Hoff, thank you for joining us today.
Thank you, Miranda. Let me add my welcome to the Celebration of Hope Gala, where I'll be introducing our next-gen investigators, and we're going to be speaking about giving pancreas cancer a nervous breakdown. For this gala, we want to feature our next-generation investigators. Some of you already know Dr. Erkut Borazanci, and he'll talk about his study, supported by this group, in patients with advanced stage 4 pancreas cancer to obtain tumor shrinkage by harnessing the patient's own immune system, made possible by this event, and his plans against the disease. Dr. Talia Golan, who you may not know, a fantastic investigator from Israel, who this year gave us a new maintenance therapy to help keep people in remission. It was a real breakthrough for targeted therapy, and she'll tell us about her plans against the disease. And finally, Dr. Doug Evans, outstanding surgeon, probably the most outstanding surgeon in the United States, pancreas cancer. He's going to speak about his study for personalized treatment of early-stage pancreas cancer to make operable patients more operable and inoperable patients operable. And that study is just starting, again, sponsored by this event. But first of all, I'd like to give you a few words from yours truly. We want to remember some of the incredible people who have been at this event in past years, some incredible fighters, because we mourn their loss. Incredible, fine people, and all of us work to honor their memory. Our mission, strategy, and tactics with your gifts are clear. Our mission is an urgency to early detect and cure pancreas cancer. But our strategy is a bit different. We want to conduct guerrilla warfare-like clinical trials based on the best possible science, usually 10 to 60 patients in a trial, and look for dramatic shrinkage in response of patients' stage 4 tumors. Then confirm those with larger studies for the FDA, which usually takes about 800 patients, and pretty long time, I'm afraid. And then apply those results to early stage patients with less burden, better chance for a cure. As a point of order, what do we mean when we use the word response? Response means more than a 30% decrease in the size of a patient's tumor. That's major shrinkage in stage 4 disease. As you can see here with these holes in a person's liver, that's the tumor. We call it a Swiss cheese liver, and before and after therapy. This is a pretty dramatic effect of a therapy developed under your auspices and support. A response, importantly, translates into a meaningful increase in survival, which is what we're after. Our tactics together are, number one, stay connected and meet with and listen to and capture the best science from all over the world. And some of the fantastic investigators that help us get these trials up and running are pictured here. And then we translate that best science into a clinical trial protocol that's doable within a budget. And then we have the very, very best clinical scientists, which I've introduced some already, perform the clinical trials as rapidly as possible, and hopefully generate the very best dramatic results to improve survival and apply earlier in the disease. Now, the only way to defeat pancreas cancer is you have to apply the best possible science against a disease. And we need to have and continue to have your help to apply that science to patient care as rapidly as possible. Our track record of working together on the mission is substantial. There's a lot of words on this slide, but on the left-hand side, we have the science concepts. For instance, number one is pancreas cancer is trapped in a scar. This is what it looks like in the middle, and to the right, the results of targeting that scar was we put together the regimen of Abraxane plus gemcitabine, shrinks 46% of patients' tumors. It was FDA approved for frontline treatment because of your support. Number two, we did molecular profiling, found a high enzyme in tumors versus normal cells. And to the right-hand side, started a regimen that gave a 45% improvement in survival for patients with stage 4 pancreas cancer. It became FDA approved, Onivide 5-FU regimen. Number 3, molecular profiling showed us that DNA in pancreas cancer can't repair itself, so it has a vulnerability. And Gail Jamieson took us into a major trial of Abraxane plus gemcitabine plus platinum. 71% of patients' tumors shrank. Some have had complete disappearance of their stage 4 disease. This is now in a larger trial. And finally, based on the 2018 Nobel Prize discovering research that showed that cancer actually puts brakes on a person's immune system, so our immune system can't identify this foreign invader. It's hidden from the immune system. With the CENA-4 trial and Mattress Firm, Dr. Erkut Borazanci, to the right-hand side, he put together a 5-drug regimen we call the Grand Slam. 83% of patients' tumors responding, and he'll also speak about intra-tumor vaccination being supported by this event. So our goal is best science into the clinic for patients as soon as possible. Now briefly, the new science concept we're working on at the time of this report today is we call giving pancreas cancer a nervous breakdown. Now there's a great deal of accumulating evidence that cancer cells are under incredible stress. This year in particular, I think we all can identify what stress is. But what stresses are cancer cells under? Number one, they've been growing under adverse circumstances for a long time. They have a lot of problems because they have a lot of chromosomes. They're so abnormal. So you usually would have 46, but the pancreas cancer cells have hundreds. Pancreas cancer takes about 19 to 23 years to show itself in a person. You can only imagine how many chromosome abnormalities, and yet the tumor cells still survive under that stress. They have a lot of scar, as we've already talked about. But what we haven't talked about, that scar gives you a very poor blood supply, low pH, low oxygen, high osmolality, so they're growing in a syrup-like environment, and very low nutritions, low sugar, low proteins, low amino acids. They have a lot of inflammation. And these tumors are under stress of chemotherapy and radiation, and yet some of these tumor cells still survive the stress. In reality, these tumor cells are under incredibly high stress, but they have to keep dividing, making the building blocks of DNA, RNA, and protein to make more cancer cells. And they have to stay alive, which means they need more and more nutrients to keep the metabolism going. Tumor cells need to eat. And as you've seen with many people, when they lose their muscle mass, these tumors even eat muscle mass. So these tumor cells are way out on a limb. The question is what can we do to saw that limb off? The new science concept we're trying to translate from the bench to the bedside this year is induce even more stress on these tumor cells to give pancreatic cancer cells a nervous breakdown. We want to overwhelm their coping capacities to kill the cancer cells in 20 seconds. And the question is, how can we do that? What's the science? Well, first of all, Gail Jamieson is trying to increase the stress of these pancreatic cancer cells by adding free radicals to the tumor environment. And to increase free radicals, you can use very, very, very high doses of intravenous ascorbic acid, otherwise known as vitamin C, not by itself, but in combination with chemotherapy. And we are seeing responses like this. The Swiss cheese liver we talked about, you can see how much that's reduced after only 9 weeks. Very dramatic response. This clinical trial continues. Just to show you how dramatic this is, many in the audience here are familiar with CA19-9. It's interesting that some of these tumors break up so fast that in that orange line it goes up, but every single person's marker goes down. You can see that all but one patient's tumors have been shrinking. That's below the line. We call that a waterfall plot. This is a very promising first step to increase free radicals to give those tumor cells a nervous breakdown. The next way to stress cancer cells is to use chemotherapy plus a ketogenic diet. In this mouse model, you can see that when you put chemotherapy together, which is the triple, we call it, Gale's triple regimen, and a ketogenic diet, KD, the blue line, that you get dramatic effect of improved survival. Long durations. This is very unusual even in an animal model. It improves survival. And the good news there is because of your support, this clinical trial has just started in patients with advanced pancreas cancer, Dr. Diane Hanna and Gail Jamieson, of the chemotherapy, the triple, which you supported, plus a ketogenic versus chemotherapy plus a regular diet. That is just starting. Stress those tumor cells. Another way to induce more stress on tumor cells to give them a nervous breakdown is deprive them of amino acids like methionine and arginine. And Dr. Hahn, who many of you know, is working on different GCN2 inhibitors in the lab, and we will bring that best into the clinical trial. There are also multiple other ways to stress pancreas cancer cells. There are things called proteasome inhibitors, which actually inhibit the garbage can of pancreas cancer cells so they choke on their own waste products. Does them good. We think this approach will cause tumor cells more and more stress so they literally give up. A final way to apply the ultimate stress to give pancreas cancer a nervous breakdown is to take out their oncogene engine. Oncogene is a cancer-causing gene that actually drives the tumors. This oncogene engine is called RAS, and it was called that because it was isolated from a rat sarcoma. I don't think the discoverers felt it would have any significance at one time, but believe it or not, it made it into our genome and causes cancer. And we have a little working mini group called the Avengers, working to take out that RAS engine. And there's incredible science from the special team using AI. That's why we call them the Avengers. And we have 2 oral agents which cause total blockade of this RAS engine. And we hope to start that trial sometime next month for patients if their prior therapies have failed them. We now have an easy blood test, thanks to your support, to measure stress in normal and cancer cells. It's called unfolded protein response. You put the cells under more stress, increase UPR. You can see here on this scale, a normal cell has some UPR. Pancreas cancer cells that we said are under some stress. And our goal here is to push the UPR up so these tumor cells die in 20 seconds. So putting more stress into pancreas cancer cells will have a greater effect on pancreas cancer cells, give them a nervous breakdown versus the normal cells. We call it a therapeutic index. Finally, an even better way to give tumor cells a nervous breakdown is kill them early, before they develop too many tumor cells and too many mechanisms of resistance, too many heads to lop off, like the Hydra. This would take much less of an increase in stress to kill them. So early detection, as we all know, is still the key. And we think the most promising early detection strategy is measuring EVs in the blood or urine. So let me just say a word or two about EVs. Extracellular vesicles, or EVs, are a terrific new tool for early detection. Believe it or not, the tumor cells and normal cells bud off these little packets of information and they circulate to other cells. We now call it the intranet of our bodies, EVs. The major effort to study these EVs for early detection is because they bud off of cancer cells and they have a unique covering and a unique content, and you can measure them in blood and urine. Easy test. What they contain also tells us what type of cancer we have. If you load them up with a tracer, they will go right back to where the cancer is because they have a zip code. It came from the cell membrane for imaging and early detection. And what's also interesting is you can actually load them with a therapeutic agent to kill the pancreas cancer, like what we call a STING agonist or IL-12. This is very, very exciting, and we're right in the middle of it. We're proud to announce that because of your support, we have a National Cancer Institute grant with all of the institutions, all the way from Japan, South Korea, many places in the United States, working on the early detection of pancreas cancer using EVs. And here's a picture of a team. Some have traveled great distances before COVID. to be with us. This is a big deal. We're working on for very early cancer interception. Just to show you how good this is the diagnostic performance in the initial trials. What you want here on these diagrams is a right angle. And you want an AUC, area under the curve, that's 100. In other words, 1.00. And you can see here the cell-free DNA to your left, and then the exosome, or EV-containing microRNAs, is 0.95. This is very promising. We have a ways to go to 1.00, but this is very promising. So I think we will have a blood test, possibly a urine test, for early detection of pancreas cancer, where we can apply our therapies earlier. Certainly, it is clearly the way to achieve a high cure rate. Finally, I want to say thank you to so many for your support of the gala, of the clinical trials, of the walking and helping, speaking to other patients. Thank you for that support. It means a tremendous amount to us. It's also that support has encouraged other investigators against pancreas cancer. And you're going to hear from some of the next-gen distinguished speakers. What can we do to excite a patient's immune system to attack their pancreas? Dr. Erkut Borazanci. If we can get patients to response, how can we keep them there? Dr. Talia Gallen, who's going to tell you how to do that. And finally, if you find a cancer earlier, what can you do? An outstanding surgeon, Dr. Doug Evans, will speak to us about his personalized approach to this. These next-gen physicians, scientists, and investigators are the very, very best. Thank you again for your support.